everybody, I'm Juliette.
Hello, I'm Maéva.
We are from the Master Mares.
Mares is a Master of Science, Conservation and Valorization on Marine Resources.
Today we are going to present you a new biotechnology
on marine organisms.
Governments of all the countries have been meeting all the week to find a solution to the microplastic invasion. In fact, because of the human activities, plastic is invading all parts of the world, starting with our oceans. It is estimated that 300 million tons of plastic are produced each year, and 50% of this plastic is only for a single use. 8 million tons of plastic are dumped every year and finish into our ocean, leading to the microplastic invasion. Today, we investigate to the possible new solution with the use of a new biotechnology. Let's have a look into the future. Hello everybody, so we are here today in this laboratory specialized into microplastic to know more about this microplastic invasion. We will talk about a specialist about the subject, so just follow me. So hello Maéva.
Hello Juliette.
So thank you for welcoming us there today. But our first question is very simple. Could we just have looked to what is microplastic? I'm sure that our listeners are very curious about it.
Yes, so let me show you. What are microplastic? There is some plastics that we see. They are often coming from waste and packaging. They are called macroplastics. They can be degraded in smaller and smaller pieces until they reach a size below 5 mm. In this case, they are microplastic and they are much less remarkable. If these small species are directly rejected at that size, for example from clothes washing or cosmetics, they are called primary microplastics. But if they are coming from degraded macroplastic, they are called secondary microplastics. In general, we know that microplastic quantity is much bigger than the macroplastic one. In practice, we are not able to measure it precisely.
Tired skin? Live your life at 100% without any skin problem. Our new mask is made for you, 100% poreless, anti-age. It will reduce all your problems. Our research on the new biotechnology based on PVT is incredible. You just have to relax and enjoy. Plastic cosmetic, 10-year revolution. Maéva, as you know, there is this kind of microplastic problem, this microplastic invasion. Could you explain to our very worried audience why microplastic is so dangerous?
We mainly know impact of macroplastic. For example, animals think that plastic are very similar to their prey, so they eat it. Unfortunately, plastic remains on their stomach and they don't eat anymore until they die of hunger. The danger of microplastics is different. Firstly, some chemical additives are present on their surface and are toxic. Secondly, some microorganisms attach and grow on them and can infect animals. In addition, microplastics are accumulated along the trophic web. For example, an unexpected victim is our seafood.
A Christmas without our delicious oyster is not possible. Don't forget to buy our new oyster, Oyster-ic. Sweet and tasty. This is so healthy. Well, that sounds pretty scary, right? But I heard that there is a kind of giant solution, like an ultimate solution to cure this microplastic invasion. Maéva, could you tell us more about that?
In fact, the name of this huge solution is a very small molecule that we call PETase or PETase.
Okay. So, what this molecule is doing?
In fact, this molecule is an enzyme. An enzyme in general in biology is just a molecule able to degrade other molecules. In our case, the enzyme PETase is able to degrade PET. And the PET as you may know, is the plastic polyethylene terephthalate. It's a polymer commonly used and in fact the most used plastic around the world.
Okay.
The enzyme is able to cut this very huge polymer in smaller molecules that we call monomer.
Okay.
And after another reaction, we obtain very small molecule called ethylene glycol and terephthalic acid. And these molecules are more absorbable by organisms.
Okay, thank you, Maéva. My big question is, how did they discover this incredible molecule?
The story started in 2016. Some researchers from Kyoto decided to investigate a company which was recycling some plastics. In fact, the soil of this company was completely contaminated by plastics. So they took some sediments, mud and waste water everywhere, in total 250 samples. They came back to the lab and they decided to do culture with this. So they put the sample on a PET. They come back a few weeks after and then found that some samples were creating a biofilm on this PET culture.
What does that mean?
The biofilm is something created by microorganisms. So it means that on that sample, some microorganisms were able to grow and to build this film. So it means that the PET is a kind of substrate for them.
Incredible! Do we know which organism can degrade this PET?
In fact, at that moment, they didn't know. So they decided to take the biofilm and to observe it at the microscope. At the microscope, they found a mix of organisms containing some cells, some yeast, and some bacteria. So they decided to identify which one of them was responsible of the degradation of the PET. To do that, they diluted the sample, but they highly diluted it, until when they put it on a well plate, only one organism will be present in each well. Then, on each well, they added the PET. And a few weeks after, they come back to see in which well there is still some PET. If there is still some PET, the microorganism is not able to degrade it. But in some well, they found no PET anymore. So they investigate that organisms, again by microscopy, and they found a bacteria.
Interesting. Do we know who is this bacteria?
Hey, at that moment, they just identified that the bacteria was coming from the genus Ideonella. But then, which species was it? They decided to investigate the genome of this bacteria and the proteome. And they found, in fact, that it was a new species.
And what is the name of this new species?
They called it Ideonella sakaiensis 201 and F6.
Thank you, Maéva. Now, the question is, how could we be sure that it's this bacteria which is degrading the microplastic?
Yes, indeed. So they put this bacteria in culture. Then they extract all the molecule contained by this culture, and they put the mix of these all molecules on the PET.
Okay.
And a few times after that, they observed a degradation of the PET, but without the bacteria. So it means that the degradation is coming from the mix of the molecule produced by the bacteria.
It's very interesting. Okay, Maéva, now we have the bacteria, we have the PETs, but what is the link between it?
You're right. At that point, we know the bacteria producing something. In fact, they put the mix of the molecule that we just saw in a chromatograph. The chromatograph will separate each molecules, and then each molecule will be put in a mass spectrometer. And the mass spectrometer is able to identify the type of the molecule. Then the researchers compared it with the database, as they found a family of enzymes that they know, but this enzyme was a new one. And because it was degrading PET, they call it PETase.
Thank you, Maéva. This is an enzyme, so does it have some special property to work?
Yes, indeed. This enzyme is active only between 15 and 42 degrees, but it's a very huge range and very useful for the water. And it can also be active between a pH of 5.5 and 9.
Okay.
The problem of this bacteria is, it's able to degrade the PET, but the PET that we use in our wrap, for example, is a very highly crystallized, like that is transparent, okay? But we see after several experiments that this bacteria is only able to degrade PET with a crystallization of 15%, which is very low and not corresponding at all at the amount of crystallization that we have in our waste.
Okay, it's not very efficient, but could we just try to put it into the water in the ocean where there is a huge pollution to see if it's work maybe just a bit?
In fact, not again, because this bacteria doesn't grow well with the salt. So it's an aquatic bacteria, but not adaptable to marine environment.
Okay, I've got it. Do you think that there is another application that we can have to the incredible PETAs? Because it sounds like an incredible biotechnology that we should use it for the microplastic invasion.
Yes. In fact, there is two things. Okay. For sure, at that moment, the researchers need to improve the activity and also to find another organism able to produce it. But at that point, you know the molecule, so you can investigate to understand which cluster of gene in the bacteria is responsible for the expression of this enzyme. And once they are identified, you can clone them and put them in another microorganism which is able to live in marine environment. And it was done.
Really? What happened? Tell me which marine environment could we integrate this incredible molecule?
In fact, it was a diatom. This microalgae is Phaeodactylum tricornutum. It's a photosynthetic microalgae, so it just needs light to growth. So it means that for an industrial application, it's very interesting because this organism required less resources.
Mm-hmm.
Then this algae, of course, is able to grow in marine environment that we want.
It's the same enzyme that we saw before, like the hydrolase. Does it have the same problem for the efficiency about the crystallization of PET?
Yes, in fact, even if we increase a bit the activity, it's still not enough to degrade the PET released in the seawater.
Okay.
But maybe the problem is solved because this week another team in another part of the world published an article and they introduce this enzyme in a yeast, Pichia pastoris.
Okay.
And where the strategy is different, that is the enzyme is not secreted in the environment,
okay,
but attached encore in the membrane of the yeast. And because the enzyme is anchored, the environment is more stable from the enzyme because the pH, the osmolarity doesn't change. So the activity is multiplied by 36.
Wow, that's incredible. I mean, and it's just this article have just been released.
Yes, just a few days ago.
That's very interesting. Thank you, Maéva, for this information about this microplastic invasion and how we can try to fight it. And thank you, public, to be there and to follow our new news biotechnology to fight this horrible microplastic invasion.